Prokaryotic cell and eukaryotic cell differs in their nucleus. In case of eukaryotic cell, a clearly visible nucleus is present. But in case of prokaryotic cell, there is no nucleus. But still, they have genome, right? So where they put their genome? In case of prokaryotic cell, their genome resides somewhere in the cytoplasm known as nucleoid. In my previous video, I already discussed about the major difference about prokaryotic cell and eukaryotic cell. In this video, I am going to discuss about the nucleoid and the nucleus, the two genomes of two kinds of cells. Hello friends, welcome to Biology Concern. I am Animesh. So let's jump into the topic. In case of eukaryotic cell, there is a clear nucleus present. In here, nuclear envelope, nucleolus, chromatin fiber, and nucleoplasm is present. Nucleoplasm is present. But in case of prokaryotic cell, there is no nucleus. Rather than, there is some region where the DNA is coiled up and it resides somewhere in the cytoplasm known as nucleoid or genophore. So you see here, in case of prokaryotic cell, we have nucleoid or genophore, that is the DNA coiled up in the cytoplasm, and in case of eukaryotic cell, a clearly nuclear envelope is present, in where nucleolus present, nucleoplasm present, and chromatin fiber was also present. So what is the major difference between two of these? That means nucleoid and nucleus. Let's check out the composition of those two genomes. In case of eukaryotic cell, 9 to 12% DNA is present in their nucleus, and 15% of histone proteins, 65% of enzymes and neutral and acidic proteins, 5% of RNA, and 3% of lipid is present. But in case of nucleoid, 80% of DNA is present, 10% is RNA is present, and 10% is protein is present. So here you observe that the major thing is component in prokaryotic genome is DNA. Here it is 80% of the DNA. But in case of nucleus, here 65% of majority of enzymes, neutral and acidic proteins, but 9 to 12% of DNA only present, including with 15% of histone proteins, 5% of RNA, and 3% of lipids. So let's talk about the nucleus first. The DNA in the nucleus packed in somewhere or with the help of some proteins like histone proteins. In case of eukaryotic cell, H1, H2A, H2B, H3 and H4 histone proteins are present, where H2A and H2B are lysine rich and H3 and H4 is arginine rich. That means in case of H2A and H2B, lysine amino acid is abundant, and in case of H3 and H4, arginine amino acid is abundant. But in case of nucleoid, there is no histone protein present. The histone proteins H2A, H2B, H3 and H4 produces or makes a histone octamer, in which DNA is wrapped around and form a nucleosome. In that way, they actually compact their DNA in a small region and known as chromatin fibers. Okay. Next thing is that about nucleolus. So what is a nucleolus? Nucleolus is a region where DNA is also compacted, and nucleolus is prominent, or nucleolus, the major function of nucleolus is ribosome biogenesis. That means ribosomes are produced from nucleolus. That means the gene that encodes the ribosome rRNA or ribosomal RNA proteins are present within the nucleolus region. In case here 28S rRNA, 18S rRNA and 5.8S rRNA genes are found in case of nucleolus. Okay, but in case of prokaryotes, there is no chromosome compaction. That means there is no nucleosome, there is no histone proteins, there is no histone octamers. But they have histone-like proteins known as NAPs. That means Nucleoide Associated Proteins. Some examples of NAPs are HU, HNS, FIS and DPS. The full form of HU stands for histone-like protein. In case of HNS, it is heat stable nucleoide stimulating factors. In case of FIS, it is factor for inversion stimulation. In case of DPS, that means DNA binding protein from starved cell. So those NAPs or nucleoide associated proteins helps the DNA to compact or to coiled up in a manner, and they are reside somewhere in the cytoplasm called nucleoid or genophore. But in case of here nucleus, we clearly observe a nucleosome that packs a DNA and produces chromatin fibers. Another most important thing is here causal bodies is also found. So main function of causal bodies is RNA processing, and causal bodies are mainly produced by a protein named as coilin, which compactly bound and make causal bodies. So coilin is the main protein that is found within the causal bodies. Next thing is pica domains. Pica domains are produced in case of eukaryotic cell. Pica stands for polymorphic interphase karyosomal associated proteins. The main function of pica are still unknown, but still pica domains are already present in case of nucleus. Another important domain, that is Pml domain, is also present in case of the nucleus. What is this Pml domain? Pml stands for promyelocytic leukemia domain. So promyelocytic leukemia domains are already present inside of the nucleus, but in case of normal cell function, Pml domain does not need for a normal cell functions. So here you clearly differentiate between nucleus and nucleoid. I think this video is helpful for you to understand the basics of nucleoid and nucleus. If this video is really helpful for you, please hit the like button and don't forget to subscribe. Thanks for watching.